Welcome back. Welcome to our team-based learning exercise. We've titled this one, indeed, as we titled the last one, Social, Ethical and Legal Responsibilities of Life Scientists. So one or two people who are here today were here last year when we did this, and so I'll skip the joke I made then about your lunch and cognitive enhancement. We've heard quite a lot about that already. I think the first thing to say is that Malcolm and I have been motivated in some ways by an article that was produced by a guy called Matt Meyerson, who you've heard a little bit about already. He's a professor of molecular and cellular biology at Harvard University, and he wears a number of hats, if you like. He's obviously a professional molecular and cellular biologist. He's also been somebody who's been involved in an interest in the science and technology and in arms control, and in that regard, an interest in weapons of mass destruction and the prohibition regimes that relate to those weapons of mass destruction, especially in regard to chemical weapons and to biological weapons as well. So in the year 2000, in the CBW Conventions Bulletin, which is Chemical and Biological Weapons Conventions Bulletin, which is produced by the Harvard Sussex program, Meyerson was considering what we were going to do with biotechnology. And the 21st century, I suppose, is the century of biotechnology. And one of the things that Meyerson talked about in his article in 2000 in the CBW Conventions Bulletin was whether or not we were at a crossroads and whether or not we would choose to use biotechnology for hostile purposes. And I think Meyerson was uniquely positioned, possibly, in regard to what he could see in terms of future scientific and technological developments. One of his observations was, of course, that we've used every scientific and technological development that we've invented, we've deployed for hostile purposes. So the obvious ones are physics and chemistry and biology. So nuclear weapons, chemical weapons, biological weapons, the internal combustion engine, all of the kinds of scientific and technological innovations you can think of, we've turned them, we've deployed them for hostile purposes. So the question in this article in 2000 was, will we choose to do the right thing and not deploy biotechnology for hostile purposes, and so will we take the right turn, as it were, at this crossroads, and choose not to do this? And so this was a number of years ago, 2018, obviously nearly 2 decades ago. I don't think Meyerson could have seen, where we would have been in terms of the mapping of the human genome right now, and in terms of the technology that we're going to talk about today, CRISPR-Cas9. But one of the things he was worried about in that article was the extent to which we might be able in future to manipulate inheritance and development and cognition and a range of other important human traits by way of manipulation of the human genome through biotechnology. So I suppose that, this is a sort of long-winded introduction to sort of explain what our motivations were. But I think CRISPR-Cas9 is a very interesting technology. It's been around insofar as the general public's awareness of it for about 5 years now. And we've seen since about 2013 this technology being deployed in regard to agriculture in particular. And so we're gonna go through some exercises today to talk about CRISPR-Cas9. And we've tried to make this as obvious as possible for the non-scientists in the room. I'm not a scientist, so I obviously need to be able to sort of understand what's going on. And I wrote this, so I think that if I've been able to develop this scenario and understand more or less what's going on, then I think that you should, the lay people in the room, the non-scientists should also be able to understand what's going on. And then hopefully those familiar with life science, life and associated sciences will be able to perhaps deploy some of their knowledge and understanding to help us better understand what's going on with regard to this particular technology. So the first thing to say then is that we just need to be clear about terminology. We went through this in a similar fashion last year. CRISPR-Cas9, I suppose we really need to sort of explain what that is. Let me just begin then with the definition. And so the acronym is Clustered Regularly Interspaced Short Palindromic Repeat Associated 9 Nuclease System. So you can see why there's been a need to sort of abbreviate that into CRISPR-Cas9. So we'll say a little bit more about that, and the idea with this exercise is that in some ways it's pretty much contingent on the preparation that you guys have been able to do. So one of the wonderful things about team-based learning exercises is, from the point of view of the facilitator, is that the students really need to prepare in advance. And of course, one of the big challenges that we've got where our students are concerned is getting them to read in advance of the class. So the thing is with team-based learning is that if you show up and you've not prepared, then there's a good chance that you're not going to get a good grade. Because the very first thing you do when you walk into the room, when you're doing team-based learning, is you do a test, which is based upon the pre-reading that you've done. So I know some of you have done this pre-reading in advance of this class because I've been watching some of you reading it from your computer screens at the 11th hour. So, but some of you are a bit slightly more prepared than that. So one of the great things about that is that the test is contingent on the extent to which you've been able to prepare in advance. So we've circulated some pre-reading, and there are 3 items that you really need to pay attention to. The first one is a very short PowerPoint presentation that I prepared on CRISPR-Cas9, which I intended to use as a very simple explanation for what CRISPR-Cas9 is in lay terms, in general terms, and what it does. Okay, so that's the first thing. The second thing is I circulated an article by Kang and colleagues, which is a slightly sort of longer read, but 3 or 4 pages, and that goes into some more detail about some of the challenges and the concerns that have been raised by this technology. So that's the second thing. And then the third thing is an article in Science that was produced by Reeves and colleagues, and that relates to the use of CRISPR-Cas9 in agriculture. So it relates to a very specific scenario that have been presented as a challenge, I suppose, to some researchers. And DARPA had asked for researchers to look into the possibility of the extent to which this technology could be used to confer changes to the genome of plants. So that's the third thing. So those are the 3 things that we hope that you've been able to look at, however briefly. So the second thing to say is that that's the background regarding the preparation that needs to be done, regarding the precise things that you need to be aware of and need to read in advance of the class. So that's the first thing in terms of what we need to talk about. The second thing is I've got a couple of slides that I want to show you, because there's a concept that we need everybody to understand, at least insofar as where we're coming from, and that's the concept of what we have come to refer to as biological security. So for the life and associated scientists in the room, you'll be familiar with the WHO definitions of biosafety on the one hand, and biosecurity on the other hand. And in addition to that, you'll be familiar with the WHO concept of bio-risk management. Okay? So those three things, we would argue, are part of a wider concept of what we call biological security. Okay? So there's been some very unhelpful duplication of meaning around the terms biosecurity in particular. So what we'd like to do, just to begin with, is just to be clear about this concept of biological security. So, as you can see from this slide, biosecurity, biosafety, and biorisk management, these are WHO concepts, and they relate, for the purposes of this discussion, they relate specifically to the laboratory. Okay? And so, in terms of this wider concept of biological security then, we would argue that those three concepts, biosafety, laboratory biosecurity and biorisk management, we would argue that they make a vital contribution to a broader concept of biological security. Okay? So biological security is like an umbrella term, and under that you'll find biosafety, biorisk management and laboratory biosecurity. So in addition to that, this broader concept is made up of a wider web of integrated and complementary elements and measures that reinforce each other. So biosafety, laboratory biosecurity and biorisk management are just three parts of a much wider concept of biological security. So one way to think about this wider concept is of a web of prevention or a web of deterrence. And one of our colleagues, Graham Pearson, who came to Bradford to work with Malcolm and I in about 1996, was the former chief executive and director of the UK's chemical and biological warfare defence establishment at Porton Down. And Graham came up with this concept of the web of prevention. And this, as an umbrella term, locates these concepts of biosafety, biosecurity, and biorisk management in the context of this much wider concept, which relates really to what goes on beyond the laboratory door. So we're not talking about what goes on in labs, we're talking about beyond the laboratory door. So this wider concept involves biosafety, biosecurity, and biorisk management, but it also involves international and national prohibitions. For example, the UK, when it signed the Biological Weapons Convention, at the same time was under an obligation to enact national legislation. So we in the UK, we've got a Biological Weapons Act, which mirrors the Biological Weapons Convention. And the same with the Chemical Weapons Convention as well. We've got a Chemical Weapons Act. So this is a national domestic prohibition that reflects the international prohibition that we've signed up to in terms of our obligations under the CWC and in terms of our obligations under the BWC. So the international prohibition regime for the bioweapons is the Bioweapons Convention, and for the chemical weapons is Chemical Weapons Convention, and the national prohibitions in the case of the UK are the national domestic legislation in the form of the Bioweapons Act, Biological Weapons Act and the Chemical Weapons Act. So that's the first thing, prohibition regimes. The second thing is that biological security, according to Pearson, involves disease detection and prevention. This is not a comprehensive list, by the way, so these are sort of highlights, if you like. The third thing is effective threat preparedness. The fourth thing, export controls. The fifth thing, oversight of life science activities. And then the sixth thing, biological security education and codes of conduct. We would argue is an important part of that web of prevention. So in regard to education, awareness raising and engagement, which is one of the things that Malcolm and I have been sort of involved in for the last few years, one of the objectives of that is to seek to ensure that those that are involved in life sciences, life and associated scientists, whether they're in higher education, academia or industry or government, are aware of what their social and ethical and legal responsibilities are with reference to humans and animals and plants. So today's exercise relates to those three areas, humans and animals, in the first instance, and then finally we'll get to this part of the scenario that relates specifically to plants in agriculture. So this is a much wider concept than biosafety. It's obviously broader than laboratory biosecurity and biorisk management. They relate to the laboratory very specifically. And you can see that these are not typical subjects that you would get exposed to during the course of your training as a life scientist. You wouldn't necessarily know about the Biological Weapons Convention prohibition regimes or the Chemical Weapons Convention prohibition regime. You may have some knowledge and understanding and awareness of detection and prevention, but probably not about export controls, effective threat preparedness and oversight. And during the course of your education, I think we've discussed this briefly yesterday, you may well have been exposed to social responsibility in the context of the treatment of animals in life science research and in reference to discussions about euthanasia, life and death, stem cell research, etc. But you may not have considered what the broader security implications of your training and of your work as a life scientist might be. So the first thing to say then is that we've got this exercise on CRISPR-Cas9. This is a TBL exercise. Some of you, I think, have done TBL already. I've talked to a couple of people who've had the experience. Maybe many of you haven't. There are various stages to this process, and we've set them out, broadly speaking, on the chart over there. But I'll just go through what we are going to be doing today with some timings. So the first part of the team-based learning approach is, I've explained what one of the sort of beauties of it is in regard to forcing students who are participating to prepare, so we've got past that stage. That relates in the first instance to individual tests that you'll be asked to do, and we call these IRATs, Individual Readiness Assurance Tests, and we're going to give you about 15 minutes to do this. So this is the idea behind this is a closed book test, as it were. You do this as an individual. You're not supposed to consult anybody else or copy from people close by either. So we're going to give you about 15 minutes for that, and then once we've completed this individual test, we'll ask you to hand the pieces of paper that you've got back to us. So what you'll need to do is, and I think that there are two things that you need to be a little bit careful about. One is that we'd like to ask you first, in the first instance, to select the answer from this list of multiple choice questions. So there's a question and there's 4 possible answers, and the answers are A, B, C and D for each question. Okay. So what we'd ask you to do is indicate by underlining or circling the answer that you think is the correct answer. Okay. So that's the first thing. The second thing is that you need to be careful when you read the question, because some of the questions say, according to the PowerPoint presentation provided, which of the following is false. But some of them say, according to the PowerPoint presentation, which of the following is true. So you just need to be a little bit careful about paying attention to whether it's asking you for true or false. So we would ask that you circle the answer that you think is correct, and then we'll give you about 15 minutes, and then we'll get these pieces of paper back off you. So we've got a much broader sort of explanation of the timings involved, but let me just start by saying that first of all we'll do an individual test, and then you'll hand the papers in and we'll take those off you, and then we'll give you some more bits of paper. And then the idea is that you do the same test again, but instead of doing that on an individual basis, you'll do that as a team. Okay? Exactly the same questions. First one is an individual test. Second one is that you do the same quiz, as it were, as a team. Okay? And that will probably take us through to about 2 o'clock, we anticipate. And then we'll have a coffee break, and then my very able assistant, Professor Dando, will be doing some calculations in your absence, and then we'll come back and then we'll have a debrief. So the next part of this involves you conducting the exact same test, but this time as a group, okay? And part of this is that involves scratch cards. So this is a little bit like doing the lottery, except you don't win any money. So I'm going to give you some scratch cards. Now, one of the things you might choose to do is give the responsibility to a member of your team the job of scratching the right answer, okay? So as you'll see here, there's questions 1 to 10, well 1 to 7, because we've got 7 questions. And then you can select either A, B, C or D for question 1, and you can select A, B, C or D for question 2, and so on, okay? So the idea with this is that for the person that's been given the task of scratching, they need to be absolutely sure that you have given them the right answer, okay? Because if you don't give them the right answer and they scratch, and you don't find that you, when you scratch, you don't see an indication that you've got the right answer, the 4 points that you could have got for selecting the right answer immediately halves to 2 points, okay? If you then scratch the wrong selection again, the 2 gets cut in half again and you get 1 point, okay? So it goes 4, 2, 1. So you really need to ensure the person that's been given the unenviable task of scratching needs to ensure that your teammates are giving you the right answer to the question. So here's a scratch card for you guys. There's one for you. So obviously, part of this discussion involves teamwork, involves cooperation, collaboration, negotiation, all the sort of employability skills that you'd imagine employers are looking for. One of those skills is how you agree on a division of labour. And this becomes particularly acute when you're deciding who should be responsible for scratching the scratch card. So I would expect to hear during the course of this part of the session the team-based learning aspect to this, the team readiness assurance test. I would expect to hear you at least discussing with each other, possibly disagreeing. You're likely to become involved in the case of Sebastian's group at the back, for example, in debate about whether there's a question there that's been ambiguously worded or there may be 2 answers that are the right answers. So a large part of this is about discussion, cooperation, collaboration, negotiation, division of labour, etc., etc. So you can kind of see the general point of what the team-based learning exercise is seeking to achieve from undergraduate students, if you could cast your mind back that far. So we're going to now do the team readiness assurance test. So we would expect to hear lots of noise from this point forward. And we'll ask you to do that until we indicate that the exercise is over. And we would anticipate that we're going to take maybe, 20 minutes to half an hour to do this. There may be more to discuss than you think. Let's see how you go on. If everybody gets to the point where you've finished, then we'll bring this part of the session to a close, and then we'll announce a coffee break. And then we'll have a debrief when you come back. So over to you guys. As teams, answer the same questions. Thank you.
So B.
Try B.
Confirm try. Okay.
Yes.
Only see your hand.
Yes.
Also, if there's a star, it means.
That's the correct answer.
Oh,
Yeah.
okay. I just thought we get stars for everything.
No, it's that. Otherwise, we have to try again. It's only 2 points.
So it's only one here that's false?
Yeah.
Do we agree on that?
Yes.
Okay, Shamin, it's only...
I need a coin. Who's got a coin?
What do you need?
Well, I
You need
got
a
a
coin?
card.
You need a coin.
Do we become millionaires if we get this?
Now it looks like a lottery, eh?
Yes, because that's in the case study. It's actually mentioned.
Yeah.
Yeah.
And so
I write this one.
Yeah.
So I think this is kind of true as well. The missile, but
Yeah,
the,
but
the
again,
metaphor is like
yes.
not perfect. So maybe we leave that one out.
I think there's no ban.
Yeah, because
otherwise we won't have all these discussions.
exactly. Yeah, and you wouldn't have
So it's
this one wouldn't be
contradicting.
true. Exactly.
Yeah.
Yeah.
Here it is. In the agricultural world, CRISPR is already used.
Perfect. Yeah. So,
Yeah.
Yeah.
so then we can.
Yeah.
So we can be quite sure about this. And
Yeah.
now about this
Okay.
one.
This has an underlying assumption that you already do germline editing, but
This
you
is
normally it's a debate and not done in many countries.
Yes.
Because
So it seems that if you want to answer to
Yeah.
B, C, D, then you have to first select
Yeah.
A. So if you do not choose any of these, or if you do not choose this one, there's no answer.
Yeah. Should we do A?
Yes,
Okay.
let's go. It seems logical to.
The first thing we're going to do is, please do wait for the microphone before you speak. Even in this session, we obviously anticipate that you've got some comments and questions about the wording of the questions, whether they're ambiguous or not, or whether there are actually two answers to these somewhat esoteric questions that we've provided for you. So the first thing we're going to do is we're going to hear from you guys what your issues are with the questions. So this is like an appeal session, and if you were at my university, then there would be the possibility that if I'd ambiguously worded a question and my able assistant here, who's supposed to have checked all of this, had checked it and had not noticed there was a problem with it, then you could appeal to me, and then your marks could be adjusted accordingly. So the first thing we're going to do is hear from you guys about what the issues are with the questions, and then Malcolm is then going to give you some feedback, secondly, on how you've done. And I've just overseen the complex calculations that he's been involved in doing this. He's going to give you an overall appreciation, a general appreciation of whether there's been any difference between you as individuals conducting this test and your performance in terms of conducting the same test as a group. So first of all, let's hear any issues with the questions. Sebastian.
We had 2 queries on questions 3 and 4. In reverse order, question 4, we believe both A and B are true, because I think what you're looking for is 4A, but 4B is any changes made in germline cells will be passed on from generation to generation. It will have economic implications if you change people's cognition, if you make them more or less likely to disease, if you make any changes that are at all relevant to the way they do work. It will have economic implications. For question 3, we thought C is what you were looking for, but we also thought that it would be potentially even easier to create diseases rather than to treat them. Especially if you know roughly the sort of genes you're looking at.
Okay.
I imagine it would be quite easier to induce a cancer than it would be to treat one. So I thought logically, if you knew the genes involved, then you could remove some of them or add some of them to treat a disease, you could probably do the reverse also to induce the disease.
Okay, so we're perfectly happy to hear. I mean, I think there's a very good rationale that you presented with regard to 4B, the economic implications of all of this, and then 3, the extent to which this could be used to actually create, medical conditions. I think that you've given a very plausible explanation. The question is, are we going to adjust your marks accordingly? And I think for the time being, we'll hear maybe what other people have got to say in case anybody sort of comes back and contradicts that, the arguments that you've made in regard to 3 and 4. So.
Yes, I agree as well. I think we can have a bit of lateral thinking. Think outside the box. So you can create as well as treat.
My assistant wants to tell you about the difference between your performance as individuals and teams now. So we'll move on to do that.
Okay, so usually what you find is that the individual level goes up when you do the group because people get chance to discuss and argue about the different cases. It's very useful to run the test, and we can alter the questions slightly to take care of the issues that have come up. But basically, the right answers in the individual tests were around about 80%, and in the group they were around about 90%. So it follows as usual that the discussions amongst the team lift the level of correct answers. I think if we adjusted for the discussions that we've had, those both percentages would go up a little bit, but there would still be a gap.
Okay, thank you. Good, so we're, I think, now ready to move on to the first application exercise. This relates to one of the readings, in particular, to the reading by Kang and colleagues. This was published in 2017 and was titled Addressing Challenges in the Clinical Applications Associated with CRISPR-Cas9 Technology and the Ethical Questions to Prevent Its Misuse, published in Protein Cell. What we've not done is, of course, we've not provided hard copies of these background readings, but we anticipate that you've got these on your electronic devices and your computers and so on. So you can therefore refer to those on your electronic and handheld devices. So the scenario then is, and we're going to do this for the next half an hour, we reckon. We'd originally planned this from about 3 o'clock till 3:30, but we're slightly ahead of schedule, and in some ways that's a good thing because we want to try and get through 3 application exercises. I suppose the point about this is that we've reached this stage in the process whereby you as individuals have conducted a test, and then you've taken the same test as a group, and we've seen that your overall performances, as it were, have increased slightly. This is what we would anticipate. And then the things that you get involved in terms of the individual test and then in terms of the group work aspect of this, the collaboration, discussion, argumentation, and division of labour and so on and so forth, that you're being exposed to I suppose the characteristics of the learning process that you wouldn't normally experience had you just been sat there listening to somebody give a lecture. So already, I think, having gone through the individual and the team readiness assurance tests, we've exposed you to some of the things that, in terms of expectations about what students should be doing in terms of their learning, insofar as UK universities are concerned at least, you've already been exposed to stuff that you wouldn't normally have experienced had you been listening to a traditional sort of didactic lecture scenario where the lecturer stands at the front and talks and you sit there and listen, which is potentially fatal at this time of the day. So we're going to move on to the first application exercise. Again, this is on CRISPR-Cas9. It's based on the Kang article that hopefully you've all read. And let me just run through this. In accordance with the reading that you've done in preparation for this class, please provide answers to the following questions A to D. And these loosely correspond with the number of groups we've got today. So as a team, this is the outline of the scenario. Discuss each question and agree on an answer to the question. Write your answers on the paper provided. So we've given you quite a big piece of paper there and some felt tip pens. And we would ask that you, as a team, discuss each question and agree on an answer to the question. Write your answers on the paper provided and be prepared to provide clarifications and rationale for selecting your answers. So the expectation is that you develop through discussion with your team members, you agree on a position, on a rationale with regard to each of these 4 questions. You write them down on the piece of paper that we've given you. You can draw a picture, you can make a shopping list. It's entirely up to you the way you want to present this. But the point is that you do this through collaboration and discussion and debate and agreement and disagreement and so on, and that we end up with 4 pieces of paper with rationales on for what you feel is the best answer to the questions. And we're going to spend about half an hour doing this, so please feel free to discuss this with your teammates. Okay, so from this list of genetic correction in Table 1, we'd like to hear what you feel is the most ethically, legally and socially problematic from this list. And I see that in terms of your poster, you have a very organised and interesting presentation. So let's hear what you guys have got to say.
So we sorted all the target genes in a new table. And from the left side on, we have humans, other animals, including pigs, rats, and mice. Then we have plants and microorganisms. And the most ethically problematic model for us would be the zygote, the human zygotes, because obviously it can be transmitted or is part of reproduction, of human reproduction, and therefore could lead to ethically and socially problematic issues.
Okay.
And that's the target gene HBB on the left corner here. Yeah.
Yeah. So just to also maybe point out for the one who didn't understand that there were 2 axes inside our table. So we considered that, so it's really centered on humans, let's say. The importance of the elements in the table are increasing from right to left and also from bottom to up. So microorganism, the first from humans is less important, and the most important one will be in this column on humans. And then inside of each, you can have also a scale of importance. Now, HBB, what is it already?
So the hierarchy goes from here to here. So this would be the zygotes. Then there are stem cells which fall up, and then immune cells, which could potentially have a great impact on humans, and then there are other tissue cells following. That's what we decided.
It might be that you have read the article much more thoroughly than we have done, but I think the problem is that they are discussing different animal models that they use for targeting specific genes, but we don't know the purpose, actually, what they are, because even if they are using microorganisms or different animal models, they're probably actually going to affect the humans as well. And so I think, first of all, we need to know what this specific gene is responsible for in the organisms that has it. And the ethical issues related to the different animal models, I think we agree to a large extent with what you say here, but we also find the zygote the most problematic one because of, as you said, the inheritance, and it can actually affect multiple generations. And then it goes to the vertebrates and the non-human primates, and eventually to the humans and the zygotes. But then we found the maize, probably the least problematic, but it depends on actually what the gene in the maize are doing and how it can spread from an ecological point of view. We also discussed plant cells because it has been shown that plant cells can communicate, or plants can communicate with each other. So that's another, we were thinking very high sci-fi maybe, but can actually plants feel? Maybe it's not so ethically unimportant as we might think.
And also in terms of the animal models used, we were a bit cautious about whether there's any legal issues involved, especially, you know, where is this animals coming from and what kind of laws do they have in the countries that they are being, you know, transported from, as regular animal laws been followed before, you know, these animal models are being used. So I think we'll need more information on that.
So I don't know if you can read it, but I'll just start with the benefits, and I think the most obvious one is that you can prevent inherited disease. Should I go over here? Oh. You should. Good. So again, you can prevent inherited disease. And then another, well, point for discussion probably would be, they mentioned in the article, what about enhancement? Do you want to call this a benefit or a disadvantage? That depends on what you want to do and what ethical stance you're going to take on it. So we left that out here, but for the disadvantages or the issues or the challenges is what you're going to do about uncontrollable multiplication, so generations down the line, and what you're going to do about those generations, they're not, you know, giving their consent. And this is the issue of informed consent, what they're talking about. Plus, tying into the consent issue, what if something goes wrong? Who's responsible for it? Is it the parents, if we talk about human and zygotes and children and offspring? Is it the parents, the doctors, the scientists? Who's liable for if something goes awry? So, I don't know, we felt like the risks are numerous. I don't know if they outweigh the benefits. That's up for discussion, I guess.
One thing would be, how do we decide about this governance? And then we can use a lot what we have talked in past 2 days about responsible research and innovation, that we do anticipation, that we do reflection, public engagement, and only then we act to decide what kind of best governance would be. We have several options, but that should be decided in a very inclusive and open and transparent manner. And Karin will tell more about different options.
I think I will give it back to you. I learned a lot about the precautionary principle. I really liked it as a European standard compared to the US. Thank you for introducing me into the details. And by transparency, we really meant that it's about, like you taught us yesterday, have the lab opens, have some opportunity to go there, to control it, because whatever at least we thought like this, whatever we are able to do, we will do. So the best thing to prevent is to have an open environment where scientists agree that you can go there and control what's going on in the labs instead of hiding what they're doing. And we also thought a lot about public engagement and what it means to make what we discussed here as people who know at least something about the topic, to engage the public, what they would need to know and how they would be able to engage in these issues, especially when it comes to the risks that are involved.
Okay, so thanks for your patience. Okay, so this brings us to our second application exercise. And again, similar sort of scenario, read the following passage, as well as the questions, and agree what the most important actions you as a team of scientists could take to address the situation described in the scenario. So I suppose the thing about this is that if you've paid attention to the reading, it will have dawned on you that this is not a fictitious scenario necessarily. Again, we we will circulate some paper for you to write on, and the request is that you write your answer to the questions on a flip chart. You need, again, as you did before, to prepare a justification for your answers and be ready to respond to questions and clarifications from the floor and from other teams and from us. So, the idea is that you are an agricultural geneticist and you appreciate the difficulty of conferring vertical modification of chromosomes in plants. It takes a long time to achieve that from one generation to the next. It can take many growing seasons. As a team, however, you're working on a novel, what we've termed a novel biocontrol agent, and you come across a technique that facilitates horizontal modification of chromosomes that allows for the editing of crop chromosomes directly in fields. You and your team believe that this has profound implications. The technique that you have developed in order to disseminate this technology in the environment is via an insect-based method of dispersal. Your technique involves a CRISPR system that is engineered to be part of a virus that allows for the modification of plant chromosomes. So in other words, this technique would facilitate plant chromosomal editing by means of virally encoded CRISPR proteins that are delivered directly to plants by aphids, which sounds a bit science fiction. And this will result, you would anticipate, in increased plant resistance to environmental challenges and or herbicides. So that's the scenario. You are teams of agricultural geneticists. You'll notice, however, from the questions that we've focused a little bit already on the chemical and the biological weapons international legal prohibition regimes. What we've not done, though, is we've not talked about NMOD, which is a treaty that relates to environmental modification. We've not talked about the Convention on Biodiversity and the Cartagena Protocol, which is concerned with the transboundary movement of living modified organisms. We've not talked about United Nations Security Council Resolution 1540 either. So, what we'd like you to do is we'd like you to focus on the regional, since we're in the EU still, just about, as far as the UK is concerned at least anyway. We'd like you to talk about the regional and the national legal and regulatory measures that you're aware of that you would need to take into consideration with respect to this scenario. So that's the first thing, because we've had discussions already about the BWC and the CWC. we think already. So please do focus on the regional, national, legal, regulatory, if you know about those, insofar as they relate to your country. That would be helpful to hear what would restrict you with respect to this scenario. So that's the first thing. Second thing is that the issue of target specificity is one that arises with this technology. And we would ask you to focus on that and how you might, in terms of your engagement with the sort of plant breeding community, how you would assure that community that your technology was safe. So in a way, 1 and 2 are sort of lumped together and 3 and 4 are lumped together is one way of thinking about how this might work. The next thing would be to do with the misuse of this technology. How would you ensure, how would you assure the general public that your technology would not be misused or used for hostile purposes, in other words? And then, so 5 and 6 are lumped together. And then finally 7, what decision-making guidance would you use in order to ensure, or at least to reassure people, to show that your research, your technology was in compliance with expectations relating to responsible conduct of research of dubious concern. So we've had some discussions about the precautionary principle already, which I think were very helpful and very useful. Number 7, therefore, would be helpful if you could focus on the contribution that professional codes of conduct and professional codes of ethics might make to this scenario. So if 7's about professional codes of ethics and codes of conduct, then 5 and 6 are sort of lumped together, 3 and 4 are lumped together, and 1 and 2 are lumped together as well. So we're going to give you about half an hour to do this one, and then same scenario. If you could write your answers on the paper provided, we'll hear what you've got to say, and we'll hopefully be able to respond to your observations. So about half an hour from now, on application exercise number 2, please. Okay, so we make it about 4 o'clock, I think, 4:30 rather. So we're going to go ahead with hearing what you guys have got to say about this particular topic. If I could just declare an interest in this, in the sense that we wrote about the military programs of the mid-20th century. Some of the big states had offensive biological weapons programs, and you know, if you look back to British activities around the start of the Second World War, and you look at the sorts of activities that go right back to the First World War in regard to chemical weapons, you can see that the British were intimately involved in developing offensive biological weapons, and shared during the course of the early years of the Second World War a lot of the information, fundamental research that they were involved in, shared that with the Americans, and the Americans ended up having a really huge offensive biological weapons program. And it focused on anti-personnel agents, and they weaponized anthrax and VX and various non-lethal agents as well. They also had an anti-crop biological warfare program, which Malcolm and I looked into, and we ended up writing about, and we ended up with an article in Scientific American way back, which was quite a novel achievement at the time, I suppose. And then subsequent to that, I ended up writing a book about the subject, biological warfare against crops. And my conclusions about this were that the Americans had developed an offensive anti-crop biological warfare program to the extent that they could reduce the production of wheat in former Soviet bloc, as it were, Eastern European Soviet satellite countries and the former Soviet Union. And they could reduce the production of rice in China to the extent that serious social disruption would have been caused. And so they were using fungal plant pathogens to do this, stem rust of wheat and pathogens that affect rice blast, more commonly known as. And so this was a very significant military program, and it's believed that the Soviets also had similar capabilities. And if you consider that Yeltsin announced that the former Soviet offensive biological warfare program lasted until the early 1990s, then and allegedly got into genetic modification of organisms, for example, with regard to anthrax, enhancing the environmental sort of stability of anthrax in the context of environmental influences, radiation and rain and blast overpressures and so on and so forth. We ended up with some very interesting information, and a lot of the information that was derived, that was used in this book, was obtained from the National Archives in the UK and the US, and you could see by examining the Public Record Office in the UK what the British were talking about with the Americans, and you could see a similar story from looking at the American archive. You could see what the British were doing, and you could sort of piece this together, along with publication in the open scientific literature. By the scientists who were working on military programs, you could see actually what was going on in military programs by looking at the civil scientific literature. So I don't know whether that's still the case now, but maybe not post 9/11. But so I sort of declare an interest in this. This, of course, is not a fictitious scenario. This scenario that we presented to you, application exercise number 2, relates to an article that was brought to our attention after it was published in Science. Some of the authors of this article got into contact with us, and they were sort of wondering about the extent to which this was potentially a biological weapon. And so we've entered into sort of discussions with them, as it were, about these issues. And so this revolves around DARPA providing finance to see if insects could be used to deliver this genome editing CRISPR-Cas technology. And so this is about aphids. So it's a real scenario, and there's quite a lot of money involved in it. 4 years over which the sort of program of research takes place involves a fairly considerable sums of money, $47 million in funding. And from what my reading of what DARPA's sort of mandate was to these research groups involved, they did not give them a mandate by any stretch of the imagination to test this in the environment. So, this was a sort of proof of concept thing. But the conclusion seems to me to be that the implications of this are that it may be possible to get aphids to disseminate to maize crops this CRISPR-Cas technology. So in theory, it's a theoretical exercise, I suppose. So there were limitations, I think, on what the expectation was as far as DARPA was concerned. So this is based upon a real sort of scenario, and the idea is that if you're a farmer and you hear that next month a plague of locusts is going to arrive, then you reach for your aphids, and they infect your maize crop with genetically modified virus programmed to insert a locust resistance gene to maize plants, and then you have this miraculous and slightly science fiction sort of approach to resilience where crops are concerned. So the idea is that this scenario is not based on a fictitious. It's not based upon sort of something that we've made up. It's based upon this agricultural research or a new bioweapon system, which was produced by Rees et al. So the idea with the scenario is that we've already had some discussions about the Chemical and Biological Weapons Convention. Of course, if this was done for hostile purposes under the Biological Weapons Convention, it would be completely banned, But I mean, if this was done, if this technology was produced in types and quantities, that didn't fall foul of the Biological Weapons Convention, and it was done, with the permission of a state involved, for example, and it was done for peaceful, protective, or prophylactic purposes, then it could be legal under the Biological Weapons Convention. It probably, however, having said that, would fall foul of the environmental modification international regulatory regime, and also in addition to that, it would probably be a problem from the perspective of the Cartagena Protocol, which is concerned with the transboundary movement of living modified organisms, this would probably fall into the category of. So the idea is that we ask you guys to really get up with your posters and give us some responses to these questions. And could we ask Group 1 to go first?
So we had a short look on Google for Austrian regulations on this topic. So the first thing which came up was the Gene Technology Act from 1995, which was republished the years after, and also talked about our Mutuality Act from 1955, and who was reinvented 2001. So it was forbidden for Austria to have chemical and biological weapons, and as well as heavy weapons which can be used for assault and attacks. And 2001 it changed a little, and so now it's just that transit is forbidden for chemical and biological weapons. If we go more into detail, I just found really a lot of regulation for Austria that refer directly to other conventions from the UN or the EC, and also, for example, the CVC regulation for Austria. Yes.
So in order to tackle the safety for plant breeders, we thought about doing long-term studies with greenhouses and not only in a small scale but large scale, and to also not only involve, for example, one institute or one company in this research, but to involve trustworthy institutes and testing labs from maybe all across the world, and universities, and some different institutes, not only companies, and for example, not only DARPA.
So for question number 4, concerning how could we ensure target specificity, the fact that this target specificity will be guaranteed in relation to the use of the technology. So there might be a lot of answers possible here. We focused on the fact that, so as stated in the articles, the CVC, for example, so it implies inspections of the different places where the chemicals are used, and so what could be done to improve all of that would be to facilitate this inspection, for example, by having an up-to-date database of our customers, and containing many informations, not only their location but also the quantity that we are selling and where they're going to use it. So when an inspection must be done, then the inspectors can compare the database to the facts quite easily.
In order to make sure that the boundaries are so that the insects can go across like whole countries or so, or will reproduce uncontrolled, we would suggest an expiration date for our insects, so maybe they should be sterile.
As for the last two ones, we think we can guide our decision making with the framework of ARIA: anticipate, reflect, engage, and act. To increase public engagement, we can use social media such as YouTube to introduce the topic with more transparency.
All right, so in our group, neither of us had any background knowledge on EU policies or policies in our countries. But Philip very quickly found a directive from the EU Commission, European Commission, from 2001 regarding the release of genetically modified organisms into the nature, so exactly what we want to do. And that's a 63-paragraph document, so it has a lot of details on what you should do, and that before you release anything you have to tell a national authority about your plans. So of course all these security measures would have to be met by us scientists. Then maybe to add to the last point from the group before, I think that these like YouTube sessions, it sounds like a good idea, but it probably leads into that direction what we discussed yesterday of kind of a one-way talk, like from scientists telling the public what they are doing. And we thought about that it might be possible to include farmers and people directly affected by these modified crops in the research process. So there would certainly be people willing to kind of give their greenhouses to scientists if they have the feeling they might benefit from it, and they might end up with well, improved crops, of course under the risk that it doesn't work and the crops die or something. But doing that, like these people would be included in the process from the very beginning, and it wouldn't be the case that the scientists come out with a market-ready product and tell everyone that's what we want to do, and now please say yes. Yeah, that's just one idea.
Right, and in terms of security, I mean, it goes in line with what you just said about the market-ready product. How can you, well, you cannot probably really ever ensure that, you know, it won't fall, or that there's some malintent. If you have malintent, you probably get your hands on this product in some way, shape, or form. So you always have this loophole of human error, we called it. But you could mitigate this issue by, for example, not only developing this product and making it market ready, but also have the servers, so people on the ground actually applying it, that at least it doesn't change hands as often, and have, just like you mentioned before, for example, to have the insects be sterile, or pretty much have them die off after a certain time space, as they suggested in the article as well. So you can really control not only who gets their hands on the product, but also what the product does and how much influence it can take and how far it can spread, right? And then also tying in what you just said and what's been said already, how to engage the public in it. Well, and this goes along with the whole security issue, is to let the public decide what they want to know and what not to know. Because if you were all transparent about it, then you probably would run the risk real fast of people misusing this knowledge. So what is it that the public wants to know, and what, you know, don't they want to know? So, but this question would be left up to them to decide. How much knowledge do we have? At which point does it get, well, say, risky, dangerous, so that this knowledge would fall into the wrong hands, so people with malintent, and have them decide how much they want to be involved in the whole transparency and knowledge thing also.
I think for us, we were keen to start with a risk assessment. We think it is best to conduct an impact assessment of what the proposed research would have. So in doing that, we'll identify potential risks that might come out from it, from the animals, plants used, and also the public who's going to benefit from it, and as well as the researchers themselves. So after identifying it, we would want to look at how to minimize this risk. So minimizing the risk would be speaking to potential beneficiaries and trying to understand what dangers it poses for them and how we can be able to help them and liaise with them. Working with professionals as well, so looking at any authorities, any licensing agencies that we need to be aware of and trying to see perhaps this has been done before and how do they manage risks in terms of conducting this type of research. And then we would also understand what international laws there are, looking at EU policies, looking at privacy, the GDPR, and trying to understand whether or not we need to be aware of specific provisions of these international laws. And then we want to also be aware of responsible research and innovation. How can we do this responsibly? How can we sort of be inclusive in this type of research so that the materials we use would fall under intersectionality and equality and making sure that we're not unduly benefiting other parties. We also want to be aware of public engagement and again, how can we align the process and the outcomes of this research with the public. So we want to perhaps, being beneficiaries, we want to make them to be involved in the process. So that would warrant us to maybe conduct some sort of awareness day where we try and engage public as to what we want to do, how this will benefit them and how they can contribute. We'll provide them with the knowledge that they need, and we are also talking about we do not want you don't want to sound so possessive of the research, so we want to give them the opportunity to go and explore. So we would provide them with knowledge base and they could find out more, they would want to be involved. So it would be that there would be some sort of commitments from their parts, and maybe this will be a good thing, and obviously we can work together, researcher involvement with the public. I think I'm going to give you to carry on here.
Yes, we were discussing the method that they were using, the aphids. They are actually, some of them are plant specific, others are not. They are generalists. And I think you have to be aware that it might actually spread to other plants. And also the aphids themselves are eaten by other insects, like ladybugs, for instance, so that can actually cause an even more widespread difficulty. And I think when it comes to these risk assessments, I don't think you actually can be totally sure that there are no risks involved in this, and also for the dual use that there is a double-edged sword, and you can't actually say that this will not fall into the wrong hands.
So at the end of it, another thing we'll be keen to do if we are successful in the research is to sort of have some dissemination events. So we want to sort of let the public, both those that were part of the research and just those that would be beneficiaries, let them see what we have done, how we've done it, and let even members of the public that have been part of the research, let them talk about what their experiences and how they've been able to learn more and how they would now go away to be better citizens maybe, or just use that for themselves. And throughout the process we want to be aware obviously of ethical considerations and ethical issues around the research and how to minimize this. So I think that's everything we talked about. Yeah.
Thank you very much. Thank you.
What we're going to do is freestyle, and I'm going to be arguing for, and these ladies will be arguing against.
Oh.
So on the international legal and regulatory measures, we even on the pro side thought that this might be in contravention of the 1972 Biological Weapons Convention due to the lack of a peaceful justification and the availability of other means to achieve the same purpose that weren't as easily weaponizable. We then looked into, since we're all EU members, we chose one of our countries at random, which was Denmark. And in Denmark, there was a ruling that the same measures or regulations would apply as other transgenic organisms, but we didn't quite have enough time to look into what those might have been. And so then we got into the areas of disagreement. So how would you assure plant breeders that your technology was safe? I would go through first the mechanistic explanation, what we thought in theory would happen, and make sure that that was tight. Then try this out in a sandbox first, where it was not possible for things to spread. After which I would try to find a controlled situation where we could do a controlled trial. Introduce it in one area and not another. And we thought perhaps we could engineer the aphids in such a way that they had a sort of fatal reaction to getting outside the sandbox, whether that be a mineral or whatever that was only available in one place, or as somebody else said, that even if they got out, then perhaps they could be engineered to be sterile. What would you say?
Yeah, I think we agreed on this point.
Yeah, that's true, actually. Okay.
We worked hard on it
Yeah.
to let them die in the sandbox.
How would we ensure that target specificity was guaranteed in relation to the use of this technology? So this was actually the same idea. We would try to make sure that they could only really survive in the areas they were supposed to. We couldn't really think of how to guarantee target specificity within that area.
Like you mentioned before, the group form, like the group before us mentioned, other animals will eat them and be a lot of risk involved.
So we go first and the next one?
No.
Alright.
You first go through the
So for the hostile purposes, we thought of first methods of cooperation, or stick and carrots. So if we could have some kind of technology exchange, some kind of agreements, and if these would be, somebody would transgress against these, we would have some kind of tariffs or other kind of intervention. That's the stick part. We thought a lot about the word ensure, which sort of implied a degree of certainty that we weren't sure we could actually guarantee. And then for the public discussions, we would also try to be, well, there's two ways to look at this. If this is really a cover for a military thing, we wouldn't tell the public that, right? That would not be clever. We would talk to the government. But if it is actually for beneficial purposes, like it's presented, then we would want it to be as open as possible, so that people can come with concerns, sort of like the open source movement. Finally, I don't think we got to what decision-making guidance would you use in order to show that your research and your technology was in compliance with expectations relating to responsible conduct of research of dual use concerns. But clearly what we would do is use all the tools that we learned about these last 2 days, such as ARIA and RRIs-tools.eu. Yeah?
So, as you can see, we worked very hard on some issues like how can we make our little viruses die in our sandbox, how we're going to do this. And when we came to the end of this list, I had this feeling, wow, this gives you really a fake feeling of security. Why not just say, just don't do it. Do something better with your time, because I don't know how you felt, but for me it was like you go through this list and you think, oh, I checked everything, and I go through all the standards, and I'm just the person in the lab, and I have to contribute to my family and to the funding of the agriculture firm. And if I do all this, I did everything well. And I involve, I do something on public engagement, I Twitter it, and then it's safe. That's that's what I think. So it gives you a fake feeling of security to go through this. And that's why I said, never forget your conscience and what you are doing. It does not free you from your responsibility, even though you have these guidelines. There are better things you can do in agriculture. Okay,
Okay,
so
and
that was the dispute then.
Yeah, and actually there were 3 different opinions. So Sebastian was more for, Karen was more against, and I was more abstaining, because I thought as a social scientist I really need more information. I really need more knowledge about all these things, about legal things, about also technology as such. And like 7th question, to show compliance, I actually have to know if it is in compliance. I can't demonstrate without knowing if it really is in compliance.
So, a couple of academics who belong to the field of education research and pedagogy, Meyer and Land, have come up with these notions that they refer to as threshold concepts. Now, these are slightly different, they would argue, to core concepts and key concepts. And they've come up with, it depends who you look at in the literature on pedagogy. Different authors have got different explanations of threshold concepts, and some authors identify 10, some more than 10, but Meyer and Land, who pioneered research in this area of threshold concepts in 2005, came up with an article, Threshold Concepts and Troublesome Knowledge, Epistemological Considerations and Conceptual Framework for Teaching and Learning, and this was produced in a higher education journal in 2005. And they arrived at the conclusion that they have identified 8 threshold concepts. Now, the challenge for us, in the absence of a national curriculum of any kind in the UK, in the absence of any kind of curriculum around which there's consensus about in the EU 27/28 countries, we've been thinking about ways in which we can identify issues in the area of social, ethical and legal responsibility in the area of dual use. And we're really struggling to try and identify what the big issues are in this area that we think that students should really know about. Okay, so what we're trying to do is we're trying to identify threshold concepts. So in the pedagogy literature, you can see that various attempts have been made to try and develop a methodology that allows threshold concepts to be identified, okay? And there are about 10, 15 different methodological approaches, and I think the consensus about these methodologies is that not one of them delivers in terms of definitively allowing for the identification of threshold concepts. So what we were going to ask you to do was ask to respond to the 8 questions that we've got here in writing. So Malcolm has got some handouts for you and some writing paper. But just to say, Meyer and Land regard threshold concepts as having various characteristics, okay? So the first one is that they argue that for a threshold concept to be identified as such, the concept should be transformative. And so there's obviously a big literature to this, but a transformative sort of a revelation in terms of your own education and appreciation and awareness of a particular issue might be a concept, for example, like the theory of relativity or the concept of evolution. You may, for example, have understood that the Earth is flat, and it's been given, you know, it's been sort of preordained, and you just find it in you know, as a sort of a concept that regards the world to be flat. But then somebody comes along and says, well, you know, that's not the case, and or somebody comes along with a theory of evolution, for example, that shows that actually natural selection is responsible for human, animal, and plant life on Earth, and how it develops and so on and so forth. And so, in a way, this is akin to opening a door or a portal to a whole set of new knowledge, which comes in some ways as a revelation. And, you know, so it's almost as though it's akin to a paradigm shift that allows you to develop a completely new appreciation of a new topic. So that's one of the examples of the way in which Mayer and Land think about threshold concepts. Another is that knowledge can be troublesome, so they've got an explanation of how troublesome works. Also that knowledge can be irreversible and integrative and bounded and discursive and reconstitutive, and they talk about this concept called liminality as well. So what we've done here is we've given you some very brief explanations of how these concepts, these threshold concepts, might relate to what we've been discussing in this session, but also since yesterday. And the first one says, what concept discussed today and throughout the course of this conference, if you like, changes the way you view responsible science? So this is a question about the transformativeness of the discussions that we've had. And then there's another one on troublesome knowledge. What concepts did you find troublesome? What, if anything, was identified today that you found counterintuitive, alien, or maybe even incoherent? So that's troublesome. So you may not be able to answer these questions, but we'd be delighted if you'd have a go. So there are 8 of them. The idea is that you attempt to respond to these with reference to what's been said since yesterday, and hopefully we'll be able to take away some ideas about threshold concepts insofar as they relate to social, ethical and legal responsibility.